Bonjour tout le monde, merci d'être là. Je viens de m'apprendre qu'il ne parle pas du tout français, puis je peux dire. Donc, bienvenue à cette conférence conjointe entre le département de biologie et les conférences de l'UBIS. Je suis Nadia, professeure au département de biologie. Merci à David qui organise des séminaires en biologie, et à David qui organise des conférences à l'UBIS d'avoir rendu ça possible. Je n'ai même pas eu besoin d'utiliser ma carte chouchou que lui m'a donnée quand je suis partie de l'UBIS. Ils m'ont permis d'organiser ça. Merci à ceux et celles qui ont pris le temps de rencontrer notre conférencier. C'est important de bien accueillir les gens qui font je ne sais pas combien d'heures d'avion pour venir nous donner une conférence. Aujourd'hui, on a le plaisir d'accueillir le docteur Karl Grieshop. Il étudie les compromis génétiques évolutifs, incluant la sélection sexuelle antagoniste et les renversements de dominance, avec une approche multidisciplinaire dont il va nous parler. Ça inclut des expériences de laboratoire, d'évolution expérimentale, la génétique moléculaire, puis de la bioinformatique, et aussi de la modélisation. Mon groupe cherche à comprendre les causes et les conséquences évolutives de la variation génétique sous-jacente au compromis de finesse, soit entre les sexes, entre des traits dans un organisme, les tissus, les stades de vie et l'environnement. Il a fait ses études de bac et de maîtrise à l'Université de Cincinnati. Il a fait son doctorat à l'Université d'Uppsala, en Suède. Il a fait un postdoc à l'Université de Toronto et un autre postdoc à l'Université de Cincinnati. Il a maintenant son propre groupe de recherche depuis 1 an à l'Université d'East Anglia à Norwich, Norwich, en Angleterre, dans la School of Biological Sciences. Il a reçu le prix John Maynard Smith en 2019 de l'European Society for Evolution Biology, l'ESSED. Il a aussi reçu le prix Benzelius de la Royal Society of Sciences d'Uppsala en 2022. Donc, je suis très contente qu'il puisse nous présenter ses travaux et les travaux de son groupe. Welcome, Karl.
Thank you. Is the mic on? We're good? Yeah, volume is okay. Thank you. Thank you, Nadia, for that introduction. I'm sure it was very kind. And thank you for coming. Thank you for inviting me. So, one of evolutionary biology's most long-standing challenges has been to explain the maintenance of genetic variance in fitness. And just to make sure that we're all on the same page, what we're talking about here is that if natural selection favors one allele, it will tend toward fixation, and when it fixes, then there's no more genetic variation for selection to continue acting upon. So how does natural selection continue to drive the adaptations that we observe in nature? We know that the per generation mutation rates are too low for the constant influx of mutations to account for all of the genetic diversity that we see in fitness and life history traits in nature. And so that means that there's some residual genetic diversity that must be explained by other mechanisms. And so the kind of leading framework for those mechanisms are different genetic trade-offs. These could be genetic trade-offs between environments or between life history traits or between the sexes, but basically concepts where fitness affects alternative alleles differently in alternative contexts. So one of those contexts is the sexes, so males and females. And so a lot of my research has to do with sexually antagonistic selection or sexually antagonistic genetic variation. You might have heard this referred to as intra-locus sexual conflict. And the basic concept is that one allele benefits the fitness of female carriers, and the alternative allele is the relatively fitter allele for male carriers. And this has the potential to balance or stabilize this polymorphism. It won't necessarily do that, but it will under some conditions. And I'll talk about one of those conditions today. But even if it doesn't balance the polymorphism indefinitely, you can still get a prolonged disappearance of the net less fit allele. So it takes longer for that genetic diversity to be eroded by drift or selection under these antagonistic forms of selection. This is a different way of looking at the same concept. So here we have a frequency distribution of human height in humans, human height in males and females. So as you know, males are taller than females. And so the gray bars are this male distribution here, and the red bars are the female distribution of height. But what you have in these dotted lines and solid red line here are the fitness functions. So that's the number of offspring that those individuals have with those different trait values, those different heights. And so it would appear that the maximum fitness for males would be to be about this tall. But the mean fitness for males is somewhere over here. And the maximum fitness for females would be to be this short. But the mean fitness for females is still over here. So there's ongoing sexually antagonistic selection to be more sexually dimorphic than we currently are for human height. So why aren't we that sexually dimorphic for human height? And the answer is presumably that there's some kind of genetic constraint between the sexes. There's some limit to being very different in height, given that something like height is controlled by largely the same genes in both males and females. If we could take one of these genotypes and express it in both males and females, like you can in Drosophila melanogaster, we could do something like this. This field has a history of coming from quantitative genetics, and so we've got lots of studies like this. They don't all necessarily demonstrate this, but many of them do, where a genotype that's been expressed in each of these dots is a genotype. You can express it in both males and females. Some genotypes are enriched for male beneficial, female deleterious alleles, so they have high male fitness, the other way around on these axes, sorry, high female fitness, low male fitness. Other genotypes have high male fitness, low female fitness, and that suggests that there's a lot of this sexually antagonistic genetic variation segregating in the population. During my PhD, I was using this seed beetle, Calisabruchus maculatus. I won't introduce it too thoroughly, except to just say we can measure fitness on this organism in the laboratory really well in a comprehensive way that I think is better than Drosophila melanogaster, and I run a fly lab now. So I'm criticizing my own field, my own model organism. This is a better model organism, so you should trust these estimates of fitness in the laboratory. I just happen to be allergic to it now. So, we, like others, found a negative intersexual genetic correlation with high male fitness, low female fitness in some genotypes, other genotypes that were enriched for many of those female beneficial alleles, male deleterious alleles, and everything in between. But that's not why I'm showing you this. What I'm showing this to you for is to show you that we've measured those fitness effects on those genotypes in just one temperature here, 29 degrees, which is what they're adapted to. That's the benign temperature for them. That's not a stressful temperature. And that's those black data points there. That's the exact same points that were on the previous plot. But when we measure those fitness estimates on the exact same lines, in the exact same way, but at a stressful temperature of 36 degrees Celsius, which is really near their thermal limit. They're barely surviving and barely able to reproduce. They're very physiologically stressed by this. Now that intersexual genetic correlation has flipped and it's become positive. And that is really profound because the way that this is actually happening is that it's these male beneficial, female deleterious genotypes that are having this presumably live fast, die young life history strategy that is not equipped to deal well with stress, that are actually over here now. And I don't have the plots to show you this, but I can later if you like. And it's those female beneficial, male deleterious genotypes that are actually doing relatively well under stress. Everybody's doing poorly, but these genotypes are doing better under stress than these genotypes are. And so having this positive genetic correlation under stress is going to help the population adapt to that novel change in the environment. It's going to help them adapt to that stressful 36-degree temperature. And so there's stored adaptive potential in that sexually antagonistic genetic variation. And so sexually antagonistic genetic variation is not just idiosyncratic. It's not just for those of us that like to study sex differences in selection. It has real implications for local adaptation in evolutionary biology that extend beyond the study of sexes. So this is kind of the status of this field. We know that sexually antagonistic genetic variation is important. We know that it's empirically evident. We've got a lot of quantitative genetic and some genomic evidence for it. But we have relatively little grasp of the total number of sexually antagonistic polymorphisms in the genome of any organism, their locations across the genome, the types of genes that harbor sexually antagonistic polymorphisms, the types of phenotypes that mediate sexually antagonistic selection. And so we're having a really hard time at really advancing our understanding of the total scope and impact of sexually antagonistic selection on evolution. In the 90s, Bill Rice did this really cool thing. Remember that I talked about that genetic constraint between males and females. So he decided he was going to design an experimental evolution study where he removed that constraint on the sexes. So using just basic phenotypic markers in Drosophila, he tracked one pool of autosomal chromosomes so that he forced them to be inherited from fathers to sons, and another pool of those same autosomes he forced to be inherited just from mothers to daughters. So those two pools of autosomes can respond to sex-specific selection without the negative fitness consequences that would be realized in the opposite sex naturally. I'll keep going through that in a couple of different versions as I explain our version of that experiment. But I want to introduce one more concept just to help convey some of the results. So that intersexual genetic constraint that I've been talking about, we measure as RMF. R is the Pearson correlation coefficient, and MF stands for male and female. So it's the correlation between the sexes. And if a phenotype is highly correlated between the sexes, it means that your genotype determines your phenotype, and you can't be different between the sexes. Okay, so a high correlation, a high RMF, means there's a strong constraint between the sexes. And the rationale for these kinds of experiments, this is a similar plot to the human height plot that I showed you earlier, is that if there's ongoing sexually antagonistic selection, so that these distributions would naturally like to proceed toward their fitness optima, toward their sex-specific fitness optima, but that they can't because there's an intersexual genetic constraint, or RMF, then we would say that there is sexual conflict. There's segregating sexually antagonistic genetic variation. And if we can break that constraint, you might see those male and female distributions diverge and become more sexually dimorphic. So our phenotypic marker for our version of this experiment was DS-red. It's a red fluorescent protein. It was on chromosome 2. Chromosome 2 carries about 40% of the genes in the Drosophila melanogaster genome, so it's not the whole genome, but it's targeting a large fraction of the genome. And we weren't able to do this in a totally symmetrical way, so there's some asymmetry to this experimental evolution study that you should be aware of. It comes with some caveats that we'll talk about later. But that DS-red marker on one pool of chromosomes, we were able to have it be inherited from fathers to sons. And it's important to recognize that meiotic recombination does not happen in male Drosophila at all. They don't recombine their chromosomes. So if you're tracking a marker that's on an autosomal chromosome from fathers to sons, you're tracking the whole chromosome. The whole chromosome is being inherited from fathers to sons. So 40% of the genome is being transmitted from fathers to sons every generation. The complementary autosomes of that are being carried by females 2/3 of the time, and 1/3 of the time, mothers are transmitting that complementary autosome to their sons. So it's kind of like taking that 40% of the genome and suddenly turning it into a pair of XY sex chromosomes in terms of its genealogical inheritance. And then we looked at the transcriptomic changes at the end. So this is another way, a couple of cartoons just to help everyone understand the kind of practical side of this. Is that in 10 bottles, each containing 100 flies for a population size of 1,000, you get all types, red, non-red, males and females, and you only let the males that are carrying that red marker through and the females that are not carrying the red marker through. So every generation, you mix them all together, sort for sex, sort for the marker, and let the red males and the non-red females through. We had 6 replicate populations, and I ran this for 107 generations, starting before the pandemic and ending after the pandemic, and it was terrible. This is another way of looking at that same thing, just in case you like to think about it this way. It's essentially one big cross, a big heterogeneous cross, hence the X here in the middle. So males carry that red marker on one of their autosomes. The other autosome in the diploid genotype does not carry that marker, so they have this DS red / wild type, but they're glowing red. Females are just wild type, and they're not. And so, again, it was 6 replicates, 107 generations. So I'll look first at some transcriptomic contrasts between females that either carry or don't that red chromosome that's been explicitly male limited in its inheritance over the last 107 generations, and red males versus non-red males, right? So that contrast represents the outcome of male-limited selection versus primarily female-specific selection in females, and male-limited selection versus primarily female-specific selection in males. And we got those RMF values that I'm going to talk about on the intersexual genetic constraint from an outside population. So if we just look at which genes became significantly differentially expressed, and I'm glossing over a lot of detail for how we did that. I'm happy to answer questions about that. And we plot the number of those genes that are differentially expressed across these different bins of sexual dimorphism. This is kind of like the human height x-axis that I had earlier, or any other phenotype that you can think of, but it's the expression levels of those genes. So whether they are expressed more highly in males or more highly in females, or expressed at the same level in both sexes. This category over here, I'm going to ask you to politely ignore because it has a small sample size and I'm not really convinced that it's enriched for significantly differently expressed genes. But this category here is interesting because it has a reasonably high number of genes, and the confidence intervals around that point estimate are sufficiently outside of what you would expect by chance. And so it seems like male bias genes are enriched for significantly differently expressed genes when we've removed that genetic constraint between the sexes. You see that same result again here when you're looking at the intersexual genetic correlation, RMF. So again, we have higher RMF values in the male bias genes generally, but we also have an even higher RMF for those that are differentially expressed. So we took these genes that are differentially expressed and we looked at their RMF values, right? So on average across the genome, they have higher intersexual genetic correlations. So how do you interpret this? The interpretation here is that these genes were already constrained. They cannot be expressed differently in males and females. And so when you break that genetic constraint by giving each sex its own kind of private genome and allowing autosomes to be inherited in that sex-specific way, those are the genes that change their those are the genes that evolutionarily change their expression over generations. And those tend to be enriched for male bias genes as well. And that's after accounting for the fact that our total hit list is already enriched for male bias genes. Okay, so they should have become more sexually dimorphic. then, right? We've broken the intersexual genetic constraints. So did we see progressed sexual dimorphism, progressed sex-biased gene expression in those genes? Just introduce another category here. We had a control population that I'm happy to answer questions about. So I'm going to speed up a little bit here. We're looking at the experimental females, males, and control males in those different colors down there, and we would have expected to see if sexual dimorphism increased, we would expect to see those highly male-biased genes become even more highly expressed on the male-limited background. So here is the red relative to the non-red. So if something is expressed more highly on that male-limited red background, it's in the up direction, the positive direction. If something is expressed more highly on that non-red background that was primarily, but not exclusively, female-limited in its inheritance, then it would be in the negative dimension of the y-axis. So it seems like our hypothesis was upheld here, where those highly male-biased genes did increase in their expression on the male-limited background, but we have it the opposite way around here. So what's going on? As I mentioned earlier, there are some asymmetries in this design, and so those could bear consequence on exactly how this experimental evolution played out. But another observation is that if I look just at the transcriptomic differences within the males between my focal males and my control males, and I look at that transcriptomic contrast within these are, if you know flies, these are the wrong flies. Oh, no, sorry, they're not. Within red, sorry, this is the slide that I changed right before you saw me start my talk. Within the red male-limited background males from our focal population versus the red males from the control population. So that should represent the difference, the change in the red chromosome gene expression in controls relative to our experimental treatment versus the non-red version of that. So the male-limited changes and the female-specific or female-limited changes. We actually see that non-red on this y-axis versus the red on this x-axis, the non-red contrast explains much more of the variation in total gene expression. So it looks, counterintuitively, it looks like we've found a lot more evolution happening on that autosome that we let be inherited 2/3 of the time through females and 1/3 of the time through males than we saw on the autosome that we gave privately to males to be inherited through, from fathers to sons. So it's kind of not the intention of the experiment, and so it bears some consequence on our interpretation. And so it caused us to think a little bit about this paradigm that we think of in this field about having distributions of trait values with fitness optima outside of those trait values and further ongoing sexually antagonistic selection to be more sexually dimorphic, and how that is sexual conflict if there's a genetic constraint. And we started thinking about how you could actually have sexual conflict in other scenarios. So here's a scenario that actually matches the data where you could have trait values, you could have phenotypic distributions of males and females, and their fitness optima could be different. But if there's a genetic constraint and females are actually selected in this direction, that's going to push males off their fitness optimum. And so there's strong stabilizing selection on males to maintain their phenotypic distribution, while there's selection on females to change it. And if you give females their own private autosomal genome in an experimental evolution study, it might actually push that male distribution more in the female direction, which could be why those male bias genes are expressed lower on that female limited background. So it's not the only explanation, but it is one viable explanation. A take-home that we can run away with from this experiment is that male bias genes with high intersexual genetic constraints seem to be enriched for those genes that we think are under sexually antagonistic selection. I forgot to set my timer. Did we start on time? And we're going to 1:30. Are we going to 1:30? Should it be a 40-minute talk, a 50-minute talk? Finish at 20 till? Yeah. Okay, so I'm going to go through the rest of this at a faster pace. It's older work. So up until now, everything that I've said has kind of had this underlying assumption that the sexually antagonistic alleles are just additive in their effects. So each allele that has each allele that benefits your sex's fitness will correspond to a linear increase in that sex's fitness. And I said that it has the potential to maintain genetic variation. The truth is that it's much more likely to fix one or the other alleles by random genetic drift or according to whichever sex is experiencing stronger selection. That changes substantially when you have a beneficial reversal of dominance, where the allele that benefits a given sex's fitness is also dominant in that fitness. Or put differently, neither allele is dominant or recessive, but rather heterozygotes have fitness values that are closer to their preferred homozygote fitness value. That substantially stabilizes this antagonistic polymorphism in a sexually antagonistic balancing selection scenario, more often than not, especially for weakly selected loci. And the reason that that happens is because the mean fitness for this genotype is somewhere in that neighborhood. The mean fitness for this genotype is somewhere down here. But when you have a dominance reversal, the mean fitness of the heterozygotes is up there. So although there's not a heterozygote advantage in the traditional sense, you have a net heterozygote advantage at the population level over time. This is true not only between sexes, but also for antagonistic selection between environments or between life history traits or other scenarios as well. So does this actually happen? We reviewed this in the broader sense of between environments, sexes, traits, etc. And it does happen. This list is actually longer. It goes just about to the floor. So we found maybe 14 or so examples where there are varying strengths of evidence for something that is consistent with dominance reversal. It's kind of hard to do a really thorough search of all the studies that support this because people find evidence for it and they don't realize that they have evidence for it. So I'll give you one of my favorite examples of this, which is from Susan Johnstone back in 2013. And there is this polymorphism that affects your horn type in these bighorn sheep. When you have the wild type allele, you have relatively higher reproductive success. That's the bighorn males. And that wild type allele is dominant so that if you're a heterozygote, you have just as high reproductive success. But if you're a homozygote for the mutant allele, you're the sneaker male and you have much lower reproductive success. However, that sneaker male has much higher survival, and a heterozygote that has both alleles has as high survival as the homozygote sneaker male, and it's that wild type with high reproductive success that actually has the lowest survival. So their whole paper was about overdominance, and they found that you get an overall heterozygote advantage. But the real reason that you have a heterozygote advantage here is because you have a dominance reversal. The wild type allele is dominant for reproductive success, and the mutant allele is dominant for survival. And so those add up quite literally to a heterozygote advantage. During my PhD, I developed a quantitative genetic method to detect signatures of dominance reversal in quantitative genetic data. It won't tell you anything about what's going on in the genome, but you can take a panel of strains and rank them in order of whose genetic variation is dominant over whose, and then you can do that in the males and the females separately. So if their rank order of which strain has genetic variation that's dominant over the other strains is a positive correlation, then presumably the alleles underlying that genetic variation are either dominant or recessive, but they are so the same way in males and females. A negative correlation suggests that some genotypes are enriched for alleles that are dominant in one sex, but recessive in the other sex. And that was what I actually found. So you can do this in the parametric or the non-parametric way. But I found genotypes that have alleles that are dominant in one sex and recessive in the other. And that's the same that's genotypes that are derived from the same population that I talked about at the beginning of the talk, where we know that there's sexually antagonistic genetic variation for fitness that actually turns into a positive genetic correlation under temperature stress and has that important evolutionary implication. So these are dominance reversed between the sexes. We don't know what that dominance looks like between temperatures, but that's an interesting kind of question that the field has to grapple with. So how does this actually happen? I was talking to some people today about how dominance reversals happen. So the short answer is that organisms can regulate their genomes in lots of different ways, and you should give them the benefit of the doubt that they can probably do this. but a more careful answer is that I have thought of one somewhat contrived idiosyncratic way to show you that this can happen in principle. So I'm going to present a model to you that uses some cis and trans gene regulatory tools that are familiar to us. If you know how genes regulate themselves, you know that you can have a gene here in pink with regulatory polymorphisms that are upstream in the regulatory site that have cis regulatory effects or allele-specific effects. That means that the expression is only going to happen on one homolog of a diploid genome and not the other homolog. And then you can have trans effects, so those could come from nearby or from afar, but they affect both homologs equally. And there was this biophysically explicit model that had come out in 2017 where they showed that if you have genes that regulate other genes, and we do, we know that, the product of the upstream genes compete in time and space for binding the downstream genes' binding sites. And because they compete in time and space, if they are bound to a binding site, they're physically blocking the other allele from doing so. And so if you are a better binder, you are going to be dominant to the other allele. Or if you are in greater allele-specific concentration so that you are just more abundant, even if you're not a better binder, but you are more abundant, you will be physically bound to that downstream binding site more commonly than the other allele, and you will be dominant by that mechanism. So there are two mechanisms that you could be dominant. We set up so that's just a biophysically explicit model. It's not evolutionary. It's just looking at how Mendelian dominance can happen from molecules. But if we take that same mechanism that they developed and we add one thing onto the front of it, which is a sex-limited regulatory stimulus, and then we put those gene regulatory networks into each of the individuals of 10,000 individuals in a population and run that as a forward time population genetics simulation where the expression level of this gene determines your fitness like so in this sexually antagonistic way, then we can let these binding sites mutate toward being better binders of male-limited or female-limited regulatory stimuli and see what actually happens. Does this little regulatory network evolve to a dominance reversed scenario that we expect as the adaptive outcome or not? The short answer is that it does. It seems to be working. This is, I should say, if I haven't said this already, this is a kind of toy model. It is published. It's kind of proof of concept. It's not a fully robust and thorough model, but it just serves the purpose of showing that you do get evolution taking and selecting for these adaptive haplotypes where the male beneficial allele is linked to a binding site that's sensitive to the male-specific regulatory stimulus, and the female beneficial allele ends up being linked to a binding site that mutated and evolved towards something that binds better to the female-specific regulatory stimulus. And so you can measure that as the correlation between those two sites. It gets stronger with increasing strengths of selection. That in turn causes those heterozygotes that have both the male and female benefit alleles being a little bit closer to their fitness optimum than they would be under that strictly additive case like I was talking about earlier. So Nadia, this is where this intersects with phenotypic plasticity, because what's happening here is essentially that these heterozygotes, by way of allele-specific expression, and I'll get to that slide in a second, are able to plastically shift their expression level of the downstream gene in favor of their sex, so in the direction of their own sex's fitness optimum. So with a given genotype, females are doing this, but males with that heterozygous genotype are doing this. So it is plasticity, but it is made possible by dominance reversals. So the real reason to go down this rabbit hole was to say, does this actually generate any transcriptomic or genomic patterns that we could detect in real data? And so it does, actually. When you have that heterozygous genotype, where you have a male and a female benefit allele, if you're a female, you express the female benefit allele higher. And if you're a male, you express the male benefit allele higher. This is just a cartoon of that data. This is the real data. So this is the log2 fold change in the expression of those two alternative alleles in the heterozygote females and males. So you get something different than 0 that's even more different with increasing strengths of selection. So can we detect this? You can with a method called allele-specific expression. Or at least you could, I shouldn't say you can detect it, you can test for it. I'm getting ahead of myself. Spoiler, sorry. So allele-specific expression works like this. You cross individuals from divergent populations, and you generate population-specific reference genomes. This is supposed to be a reference genome with little tick marks representing different sites of variable polymorphic sites in the genome that are fixed differences between population A and population B. You cross them, and you look at those hybrid males and hybrid females. And in your hybrid males and your hybrid females, they have reads in their transcriptome that come from autosomes that they inherited from their moms and autosomes they inherited from their dads. And you're going to try to disentangle which reads came from which autosome. And you do that by mapping every read to both genomes. You map it to the population A genome and the population B genome, and whichever one it maps to better, it presumably was inherited by whichever parent came from that population. So you do the reciprocal cross so that this is not confounded with sex, and that way you can identify the reads that were from the mom and the reads that were from the dad in the males and in the females, and those read coverages are estimates of expression. And so that would look like this if you were to just plot the expression levels of males and females for each specific allele. Does that make sense? So these cartoons are, again, not real data, and I'm going to show you a series of cartoons that look like this that are not real data. These are meant to be conceptual representations of allele-specific expression for individual genes. Of course, I'm not going to plot that for an individual gene 14,000 times. This is just a really narrow result that comes from a much broader paper that was actually just came out recently in GBE that was led by Prashasta Mishra. I was a co-supervisor on this project, and there are lots of interesting results in there that we unfortunately don't have the time to talk about. But we did look for reversed patterns of allele-specific expression between the sexes to see if we could detect that dominance reversal transcriptomic pattern in the data. So using just the whole body data, what we did was we first tested for significant allele-specific expression in just the male data. So it doesn't matter which population, it doesn't matter which allele is being expressed more highly. It's just whether or not the gene shows either this or this. And about half the genes in the genome showed that in the male data, and about half the genes in the genome showed allele-specific expression or cis-regulatory genetic variation, as you might have heard it called, in females as well. And so now the question is, what is the fraction of those hits that show opposite alleles being expressed more highly in males versus females? So a scenario like this, where allele A is expressed more highly in males, allele B is expressed more highly in females, or vice versa. But any of those scenarios where the same allele was expressed more highly in both sexes don't count for this test. And so we actually found a total of 176 genes, with as many as 26 of those being false positives. So at least 150 of those are true positive dominance reversals for gene expression between the sexes. We don't know if any of those genes have anything to do with fitness or phenotypes that are important to fitness. We just know that they have this really interesting conspicuous pattern of reversed allele-specific expression between the sexes that is consistent with sexually antagonistic selection, or the adaptive outcome that sexually antagonistic selection would have on the transcriptome, according to our biophysically explicit model of gene regulation. So 176 genes is not a lot of genes. You might think that that's a puny, unimpressive number of genes. I think that's a really interesting and exciting number of genes. I wouldn't want too many genes. Anyway, this is a kind of new territory to explore, where we've got this bottom-up approach of scraping the transcriptome for candidate sexually antagonistic polymorphisms that we could go and validate or tackle from other directions to continue advancing this field forward and try to understand more about sexually antagonistic genetic variation. So dominance reversals do happen, and we think that allele-specific expression may be a viable mechanism to find them. If you like this kind of work, I have a PhD student opportunity in my lab right now. The deadline is January 8th. Please spread the word about this and have people apply. I really like it in Norwich. It's over on the east side of England. It gets less rain than the rest of England. Most of the weather systems are kind of depleted. By the time they get over there, it still rains almost every day, but it's less rain than the rest of England. And we are looking at doing a lot of the similar kind of, a lot of stuff similar to what I've talked about here, except not just between the sexes, but also dominance reversals between environmental variables and how those two things intersect, and using genomic and transcriptomic tools, as well as some laboratory experiments to validate our findings. I have a lot of collaborators and funding bodies to thank, and I thank you for your attention, and hopefully there's time for questions.